Hi class, let's finish up our presentation for Chapter 6 with a discussion on the mathematics behind bacterial growth and how we can plot it and count the numbers of bacteria in culture. Bacteria, generally speaking, reproduce through a process known as binary fission. Bi meaning two, and during binary fission, one cell will become two cells. A parent cell will get larger and larger and larger and large and large and large until it's split in half and pop! Two daughter cells or two baby cells come from the one large parent cell. During this process, as the parent cell is enlarging, it needs to duplicate its chromosome. It could be a circular or a linear chromosome in the nucleoid region that's being reproduced. It needs to pull its cell envelope or its cell membrane together and pinch it off in the middle. And then the cell wall needs to grow down the middle of the bacterium. And once that cell wall is finished growing down the middle of the bacterium, it forms a septum or a barrier separating the two soon-to-be separate daughter cells. And once that process is finished, pop, we have the two daughter cells. So we start off with a baby cell or a young cell, so to speak. It grows bigger and bigger and bigger. It's duplicated its chromosome. It forms that protein band in the middle that will begin to initiate the septum formation, and then boop, pop at the end. We're all done, and we have two daughter cells. Now, because we start with one cell and end with two cells, we've doubled the population. Bacterial cells grow through a round of doubling. We can start with one cell, and one will become two becomes four becomes eight becomes 16, and then 16 to 32 to 64 to 128 to 256, so on and so forth. Double. This generation time, or doubling time, is the amount of time it takes to go through one round of binary fission. A generation is going to always increase a population by a factor of 2 because we keep doubling our populations through binary fission. And this process, this generation time or doubling time, is going to assume ideal growth conditions, ideal environmental conditions, so we can have that consistent doubling time. As environmental conditions change, this generation time is going to become longer and longer and longer until eventually the total number of living cells plateaus off. Now, this rate of population growth, or it can be measured via generation time. The generation time of an average microorganism is going to be between 30 and 60 minutes. The shortest generation times can be as low as 10 to 12 minutes. So every 10 minutes you double the number of bacteria or microorganisms within that population. Some microorganisms have notoriously long generation times. Genus Mycobacterium has a very long generation time. So when I would work with students in the lab and we'd have Mycobacterium tuberculosis samples, I would leave those in the incubator for a week to get a nice, good colony growing, because the generation time can be days for some of those Mycobacterium species. Mycobacterium leprae, for example, has a generation time that can be as long as 1 month to double the population. If we look at environmental bacteria, or bacteria in the environment, they have generation times that can be measured in months. These bacteria are nearly dormant. They have almost no growth because the environment is saturated with bacteria that are in antagonistic relationships with each other. Most pathogens have relatively short generation times because they need to compete with an immune system that's actively trying to kill off the pathogen. So here's a good example of doubling. We can have one cell, then two cells, then 4, then 8, then 16, then 32, 64, so on and so forth, And it's going to be a nice sharp curve if we were to graph it with a straight, boring, linear y-axis. But because if we were to graph any reasonable number of generation times, like 20 or 30 generation times, it would just be a line straight up on your graph. It's very hard to get data from that line. We typically like to go with a logarithmic scale, a log base 2 scale. And if you use a log base 2 scale, where'd my mouse go? Come back, mouse. There we go. There's the mouse. If you use that log base 2 scale, instead of having that really sharp exponential curve, you can have a nice linear curve on your graph. In a log base 2 scale, you have a steady increase in your y-axis as opposed to a crazy high increase in your y-axis. So, if we were to graph or plot using an equation to predict the number of bacterial growth or bacterial cells, we can use a log-base 2-based equation. This equation is going to require first that you know the number of starting bacteria, and we have a 2 here that represents the doubling for every generation, and then n represents the number of generations. So let's say I start off with 1 bacterium and I go through 2 generations. I would plug 1 in for n sub I. 1 times 2 to the second power would be 1 times 4. If I started with 1 bacterium and I went through 2 rounds of doubling, I would have 4 bacteria at the end. If I started with 3 bacteria and I went through 3 generations, 2 to the third power would be 2, 4, 8, and then 8 times 3 is going to be 24. So if I started with 3 bacteria and I went through 3 generations, I would end up with 24 bacteria at the end. You plug and chug your variables with this equation. If I plot the total number of viable or living bacteria, I can make a growth curve. And this is a predictable pattern of bacterial growth that looks at the number of living or viable bacterial cells within the culture medium. So it looks at the concentration of these bacterial cells. You typically start by placing a tiny number of cells into a sterile liquid medium or liquid environment. Usually this is going to be nutrient broth. And this is known as inoculating the sample. After inoculating the sample, you put it in an incubator and you culture it over a period of several hours. Initially, there's going to be a very low concentration of bacteria, and over several hours, or over those several hours of incubation, during regular intervals of sampling, you find that the bacterial concentration starts out really low, stays really low, and then all of a sudden starts to get really high and explosively grows super duper high, super duper quickly, and then plateaus and levels off, and eventually will start to get lower again. To count the number of bacteria, you can plate each sample onto solid media, or you can use specialized equipment to directly count the bacterial species in the sample. So you start off with taking a sample and you inoculate your initial sample. Initially, before you inoculate, there's no bacteria. CFU stands for colony forming units. If there is a live bacterium that you put on the plate, that live bacterium would form a bacterial colony. So one colony is counted as one live bacterium. So here we have 2 colonies that have formed. So we had 2 CFUs. So we assumed that there were 2 live bacteria that were put onto this Petri plate. And based on the dilutions that we went through, we would estimate that there were 10,000 viable or actively living cells in the tube or the flask at that time based off of the dilution ratios. If we sample every 60 minutes, you'll find that the population will start to increase very slowly at first, and then towards the end increases very rapidly. This lag phase corresponds to that very slow increase in the population growth. One thing that contributes to this lag phase, or this initial slow start to bacterial growth, is the fact that our newly inoculated cells need some time to turn on the cellular machinery that's associated with actively reproducing the cells through binary fission. These cells have been in a dormant state, and they aren't focused on bacterial or cellular reproduction. They're focused on staying alive. But if you put them in that favorable environment, they'll shift gears and shift their focus from staying alive to reproduction. During the exponential growth phase, or the logarithmic or log phase of growth, we have most of the cells in the culture that are focused primarily on reproduction. So there's lots of binary fission taking place. During this exponential or log growth phase, the doubling time or generation time is going to be at its shortest. After our log phase, we have the stationary growth phase, or the stationary phase. At this point in time, the addition of new cells equals the rate of cell death. So cell division is starting to slow down. Cell death has picked up to the point that's equal cell division, so the total number of living bacteria in solution remains constant. This is caused by a depletion of the nutrients that are in the solution. So now there's been lots of competition, and these bacteria that are competing with each other for a limited number of nutrients, generally speaking, start to secrete organic acids or other antibiotics or other chemical compounds that try to kill off their neighbors so that they can use the nutrients that are available. After the stationary phase, we have the death phase. This is when cells die off faster than they are reproducing or being birthed. And this is an exponential death phase due to the buildup of excess waste products within the growth medium. The speed that this death phase occurs at is largely dependent on how resistant a specific species is and how toxic the conditions are. If you have if you start with a known strain of antibiotics-resistant drugs, or bacteria, I should say, those bacteria are generally speaking going to have a more prolonged death phase because they are going to be resistant to the antibiotics that are naturally secreted by bacteria in the stationary phase. This death phase is going to be much slower than the exponential growth phase. It's going to be very prolonged and protracted. And then finally, at the very end, when the population levels out, bottoms out after the death phase, we're at the viable non-culturable state, the VNC. This is the state where the cells are still technically alive. They haven't died yet. There still are some living cells, but these cells are in deep hibernation. And if you put them on some culture medium to try to grow them and see if you can get them to reproduce, they won't do anything. You'll miss them, and you will assume that there isn't anything living at the time. It takes very special chemical environments, very special environmental conditions to take a viable non-culturable cell from that VNC state and bring it back to an actively growing, actively vegetative cell. So we'll start off, if we look here, we have our lag phase. We have a very low concentration of live cells. Then we have our exponential growth or log phase. Then the stationary phase. We have high concentrations of living cells. But once we hit the stationary phase, we start to have dead cells building up in our medium. And then during death phase, the population of dead cells is increasing very rapidly, and the population of living cells is decreasing very rapidly. At the very end, some cells are still going to remain viable, and that viable non-culturable state. So why do we care about growth curves? Particularly you, as aspiring healthcare providers, why should you care about this funny graph and that equation that I showed earlier? Microbes that are in that exponential growth phase are very vulnerable to antibiotics and heat. If they're focused primarily on reproduction, they don't have the genes turned on that make them resistant to antibiotics. They aren't going to have those genes turned on that make them resistant to stressful environmental conditions. And when you expose actively reproducing cells to stressful environmental conditions, e.g. lots of antibiotics, those cells are going to be much more likely to be destroyed. Actively growing cells are very vulnerable because they are actively taking in lots of extracellular chemicals into their intracellular environment because they're trying to grow and swell as quickly as possible. And they're much more likely to bring those antibiotics inside of their cells to then disrupt their cellular metabolism. Somebody who's actively shedding bacteria is typically going, in the early and middle stages of infection, is going to have lots of cells in that logarithmic growth phase, and they're going to be much more likely to spread the infection from person to person compared to a person who's in that later death phase. During the death phase, bacterial cells, generally speaking, aren't focused on reproduction. They're focused on staying alive. And a person that has their bacterial cells in the death phase aren't going to be shedding cells that can grow very rapidly. How can we count the number of cells that are in a sample? Well, we can use, first, we need to have that sample. And we can use a chemostat. A chemostat is a growth chamber, essentially, for bacterial cultures. And these are automated growth chambers that have a steady stream of nutrients going in and a steady stream of waste products going out. Chemostats are used to commercially produce antibiotics and are used to lock in microbes at constant environmental conditions by regulating the flow, having a constant stream of nutrients going in and a constant stream of waste products going out. You can maintain and stabilize the external environmental conditions. Chemostats, generally speaking, are fairly expensive, so you'll see them in industrial applications where you need to have incredibly large vats of microbes produced, or in research situations where you need to have very finicky or very precise environmental conditions constantly maintained for those microbes. To count the microbes, you can use turbidity. Turbidity is a measurement of how cloudy the solution is. Turbidity counts, generally speaking, do not differentiate between living or non-living cells. They just measure the concentration of total cells. Turbidity measurements are done by shining a light through the sample, and then that light is shined onto a photodiode, which will then measure the intensity of the light that's been shown through the sample. If lots of light makes it through the sample, there's fewer bacteria blocking the light, and there's a lower population size of bacteria. Now I want to emphasize again, the turbidity counts do not emphasize if they are living or non-living. We can do direct counts or counting techniques. Direct cell counts involve using a microscope slide with a grid. Direct cell counts are laborious, they're tedious, and do not differentiate between living or non-living. You can use a culture counter. A culture counter is going to take a steady stream of liquid media through a tiny pipette and will individually, with an electrode, scan each bacterium that goes past. Culture counters, generally speaking, do not differentiate between living or non-living bacteriums. The gold standard for counting a bacterial population size is a flow cytometer or cytometer. It's very similar to the culture counter, but is specialized so that it can differentiate between cell sizes and living and non-living cells. Here is an example of a culture counter. We are going to have a very tiny capillary tube that the bacteria flow through, essentially single file. And as those bacteria are moving through an almost single file, every time they block the flow of light to the photodiode, we will initiate another tick on the automatic counter. Another method for counting population size is with genetic protein. We can use something known as real-time polymerase chain reaction that quantifies that measures the concentrations of specific strands or lengths of DNA, and by measuring the concentrations of specific fragments or lengths of DNA, you can quantify the concentrations of bacteria or other microorganisms present. If you have high concentrations of bacterial DNA, you must have high numbers of bacteria. If you have low concentrations of the bacterial DNA, you must have low concentrations of the bacteria. So, concept checked. Put the steps in order for the bacterial growth curve. So, what goes first? And what goes last, and what goes in the middle, from beginning to end. If you do not remember, rewind the video, flip back in your PowerPoints. 1, 2, 3, 4, 5. The first stage is the lag phase. After the lag phase, we have the stationary or the exponential or the log phase, and then we have the stationary phase, and then finally we have the death phase. So the correct answer is B, C, D, and then A. If you have any questions, please shoot me an email or post them on the discussion board. Happy studies!